Annie leads with a model of partnership with patients and caregivers in which lived experience, expertise, and community priorities shape scientific, clinical, and regulatory development. Our other speaker today is Patrick Gonzalez. Next slide. And Patrick is a PhD that currently serves as VP of Clinical Sciences at Solid Biosciences. He is a muscle biologist by training, primarily focused on the molecular biology and physiology associated with skeletal and cardiac muscle in DMD. And since joining Solid Biosciences in 2016, he has led non-clinical pharmacology, translational, and clinical bioanalytical and functional analysis efforts, collaborating closely with academic research, clinical, and industry partners. Next slide. So we're really excited to have the both of you with us today. I do want to say to everyone that we're going to be hearing a lot of amazing information. If at any point you do have any questions, please type them in the chat box and one of us will read them at the end. And if for whatever reason time is not on our side, then I do encourage you all to contact me directly with any questions not addressed, and I can pass those to our presenters. Next slide. Oh, actually, we'll go back one, and I'm going to turn things over to Pat and Annie.
That's great. Thank you so much, Jennifer, and the entire team at Defeat Duchenne Canada. Obviously, thank you so much for your partnership and, of course, for the invitation for us to come and share an update on SGT-003, which is our next generation gene therapy investigational clinical trials that are underway for Duchenne muscular dystrophy. As Jen kindly, that was such a lovely introduction. Thank you so much. As she mentioned, I'm Annie Ganote. I'm a co-founder. I lead patient advocacy, and I'm a Duchenne mom. So it's a real pleasure to meet with you all today. And I'm joined by my colleague, Pat Gonzalez. So if we go to the next slide, Pat. As a publicly traded company, we may be making forward-looking statements. Our lawyers make sure that we include these in all of our presentations. Next slide. As Jennifer kindly shared, for those of you that may be newer to the community or unfamiliar with Solid, we were founded in 2013 following my son's diagnosis of Duchenne. What started off as a deeply personal mission has grown into a company that is advancing multiple clinical trials for both neuromuscular indications and rare cardiac indications, of course, with Duchenne being our lead program. We are based just outside of Boston, and we are a lean organization with about 130 employees strong, of course, with deep roots and connections to the Duchenne community. So thank you so much for this. So I'm going to pass it over to Pat, who will share an update on both our Inspire and our Impact Duchenne clinical programs, where we are. And of course, we'd love to hear from all of you. So if you want to drop questions as we speak into the chat, we will do our best, of course, at the end of the presentation to address all questions. So thanks so much for your time and your attention. Pat?
Perfect. Thanks, Annie. And just echoing that, a huge thanks to everyone for being here. And it's really a privilege to be able to go through an overview of where our program is, some of the background related to it, and the studies that we have both been running and have recently started as we really advance the understanding of SGT-003 and really looking to characterize both the safety and the efficacy as we move forward with this program for Duchenne. So I'll start with a bit of an overview of what SGT-003 is. And so what we consider this is our next generation gene therapy that we are developing for Duchenne. It is a microdystrophin gene therapy that uses an AAV, which is an adeno-associated virus, to deliver a version of dystrophin to replace what is missing in Duchenne, which is a functional copy of dystrophin. So when we look at the different components of what make up this approach, we like to highlight these three main pillars that really summarize why we consider this both a next generation approach and also emphasize the pieces that we focus on, both when we're putting together this specific therapy, as well as what we'll also be analyzing when we go into the clinic. And so in the first part here on the left is highlighting the optimized transgene that we deliver. So, as I mentioned, in an AAV gene therapy, what we are doing is actually delivering a transgene or delivering a gene to replace something that isn't functional in the body. And in the case of Duchenne, that's dystrophin. So the dystrophin is always going to be impacted on a functional side, meaning that it's not able to both have the molecular functionality and it's also not able to fulfill those roles of things like stabilizing the membrane, which then leads to the overall disease moving forward. The microdystrophin that we selected for SGT-003, like all microdystrophins, is unique. And the one that we use includes what's called this NNOS binding domain. And why that's important is really because it can be able to restore an additional piece of dystrophin that could actually help protect muscles better by getting more blood flow to the muscle. And in a follow-up slide, I'll show you how that compares to our full-length dystrophin. As we're using this specific approach, which is an AAV delivery mechanism, what we focus on as well is the actual capsid that we use here. So a capsid is actually this delivery vehicle. It's the way that we're actually going to be administering our dose of this therapy. And then that capsid is going to allow for the transgene, our microdystrophin, to go throughout the body and really get where it needs to be expressed so that it can be able to restore functionality because dystrophin is missing. So the specific capsid that we use is quite unique in SGT-003. And this is because we understand that there are naturally occurring AAVs. Some are better at getting to targets that we need for Duchenne, such as skeletal and cardiac muscle, but they're really not as good as you could potentially be, and they require very, very high doses. So when we developed this program, what we did was really take a step back, look at how we can improve upon the capsids that are already existing, and try to find a way to actually get more to where it needs to be, which are across the body, skeletal, cardiac, muscles. And then can we also reduce the amount that is getting to tissues that we don't need it to be in for Duchenne? And that's the liver. We know that over time, this is something that could potentially be beneficial both from an efficacy perspective. So we can be potentially more effective when we get more of the drug to where it needs to be. And there's a potential for this to also be safer if we can get less of the drug to where it doesn't need to be. And that's really where this capsid came from. The third piece that I'll highlight here is how we focus on manufacturing our drug and really to call out that it's very important to improve on and maintain purity of our drug. So we are still delivering a high amount of our AAVs to be able to elicit the response that we're looking for. And in doing so, when we make a lot of drug, it's very easy to actually lose the purity profile. And so that's a negative thing. What we did was actually, again, look at the process that we were using and try to find ways to actually maintain purity so that we can also be able to lower things like empty capsids, which could contribute to an overall immune response. And so we're happy to say that we have implemented this process from the very beginning of our program with the ability to then take it through each of our clinical trials as we move towards those next phases of development for this SGT-003. So again, major highlights, getting more to where it needs to be, more to the muscle. This is both skeletal and cardiac and less to the liver here, while maintaining a pure profile with a unique transgene. And I mentioned that SGT-003 microdystrophin is unique. So here's a quick visual to be able to compare that to full-length dystrophin. And this is important again because dystrophin is such a large protein and such a large gene, so that with our AAV-based approaches, we actually can't package that full-length dystrophin gene. So we need to make choices on what is going to be included in a microdystrophin. All microdystrophins are different because these choices can be made in one way or another to include some or different parts of the full-length dystrophin. There are certain components that are absolutely essential to be included. And that's when we look at this portion here, which is called the actin binding domain. That's going to be on the intracellular side of our dystrophin, where we're actually going to be binding to actin. And then here in our cysteine-rich domain, where we have another binding domain that's going to be against beta dystroglycan. So what this does is actually form the structure of the muscle. And having these domains is absolutely critical to be able to actually have that muscle membrane integrity. In addition to those, though, we have many different repeats as well as these flexible pieces called hinges throughout the microdystrophin and in full-length dystrophin. And so what we did was also choose the portions that could be the most functional. And you'll see here highlighted is R16 and R17, which are two spectrum-like repeats that actually have a unique function. Whereas many of the different pieces of this rod domain, these different Rs, can be redundant to one another. So you don't actually necessarily need all of them to do the same function. This piece is unique. And that's the reason why we wanted to include it in SGT-003, because when nNOS binding domain is present, we then have the ability to actually localize, to actually be able to bind another protein that needs to be in the correct location for it to be functional as well. And that's what's called nNOS. And this leads to the ability for a muscle to get more blood flow when you have an exercise or an energy demand. And if you think about it the opposite way, if you don't get enough blood flow when you have exercise or energy demand, you could actually cause damage. And so that's the reason for why this could be conferring an additional functionality and an additional protectiveness of the muscle when this is included. And that was a key reason for having this piece in our SGT-003 microdystrophin. So overall, to date, our clinical development plan for SGT-003 includes two main studies that I'm going to be highlighting, both an overview, giving some data on today, and then also telling you where we're moving with each of these studies as we go forward. The first study is called Inspire Duchenne. And so this was our first in human phase 1/2 open label trial. This means that this was the first study that we went into patients with, the first study that we went into the clinic with for SGT-003. And we wanted to really focus on things like looking at safety and tolerability because this was the first time we were looking at this in patients. And then also we wanted to see how can we actually start to be able to evaluate the efficacy of the drug. So things like looking at biomarkers especially have been a key highlight of our program, and we'll go through a few of those here today. Our second study that I'll highlight here, which is now also active and enrolling, is called Impact Duchenne. And so, whereas Inspire Duchenne really was focusing on looking at early data, looking at safety, and really doing that throughout a wide patient population, Impact Duchenne is focused on really looking at the functional efficacy of SGT-003. So, giving it that test to be able to see is SGT-003 ultimately doing what it's supposed to be doing, and that's going to be in improving muscle function for our boys who are being treated with this drug. And so we'll look at this study as well in the next slides. So first I'll start with Inspire Duchenne. As I mentioned, this is a phase 1/2 study. And we're very happy to say that this is a study that we've been now running since dosing began in the middle of 2024. As we got started with this study, we really wanted to be very cautious. We really wanted to make sure that we were being safe in how we were progressing. So we had significant monitoring that we did, really working very closely with our amazing clinical trial sites. We started the study in the United States. We actually quickly moved into Canada, so we're very happy to have been working with Sick Kids here for this study. They've been an amazing partner, and we'll continue to be working with them. We also have expanded the study to Italy as well as the United Kingdom, as we also wanted to make sure that we're having an impact on as many patients as broadly as possible. As of a data cut of May 12th, 2026, we had 47 patients who have been dosed in the study. And so we'll go through some of the specific details of who these patients are, but really encouraging from our side to be able to have gotten to this amount of patients dosed, because it really gives us a good idea of what we're seeing, not from just a few patients who are in, but really looking to things like consistency across a group, and especially over time, seeing how things may change. And so to date, we are nearing the completion of the planned study dosing for patients in this age range, which is less than 12 years. As I mentioned, this is an open label study, so it means that everyone who is in the study did receive SGT-003, and this is by a single intravenous infusion. Since this is an AAV gene therapy, you only need one dose. That's where we're at today. And the reason for that is because this is going to be something where once it gets into your muscle, where it gets to where it needs to be, you'll be able to express that gene over time. So to date, this is still a single dose gene therapy approach. During the study, as I mentioned, we really focused on safety. And then when we look at biomarkers, one of the big things is microdystrophin expression. So we'll actually look at that in muscle. And then we'll also start to look at function. And these will be things that take a little bit longer to evaluate. So over time, we'll be able to include some of these results as well. So of the patients that we had dosed as of this data cut, and we actually had 46 here for a safety data cut here, so we can look at each of these percentages for treatment emergent adverse events and treatment related adverse events. We're very encouraged again to have dosed those who have been in quite a wide age range. So as young as 1 year old and as old as 10 years old in this study. And then across a weight range of about 10 to 40 kilos as well. In this study, we've had one serious adverse event, so we'll call that out. And this was an immune-mediated myositis. And you may have heard about what a myositis can be. So I just want to explain it very quickly. And it's that it's a reaction to the actual microdystrophin that's being made by this approach in specific patient who had a deletion of that part of the gene. And so if you have a deletion in parts of the gene that are in the microdystrophin that's being expressed, you have a potential to have an immune response against it. And so as a result of this event, we actually have further refined our exclusion criteria to mitigate this risk for the future. However, we've been very encouraged that this specific boy was able to be responsive to treatment quickly. He didn't have any muscle pain or weakness, which actually can happen with a myositis, but that didn't occur here. And this actually did resolve soon after. So very encouraged on that side, but always being very conservative from a safety monitoring standpoint and how we look at everything within our trial. Overall, similar to what we may expect for other gene therapies, what we really see most frequently are going to be things like nausea, vomiting, and decreased appetite. And these are things that happen really within that first week or so post-dosing. And then after that, really reducing and going away. And within this study, what we focus on is using an immunomodulatory regimen. So this is something that we use prophylactically to be able to mitigate the risk of adverse events. And this is just going to be increasing steroids. So you may have heard of other programs where there are different things that are looked at, including different agents, different drugs for the purpose of modulating this potential immune reactions. We've been encouraged again to be able to stick with this specific approach for our program, and have been again happy to see the results that we really haven't had any additional side effects that might require additional agents as well. So when we look at the biomarker data, now kind of switching from safety now to some of our efficacy evaluations here, the first thing that we looked at is actually going to be in the muscle. Because SGT-003 is delivering our transgene to muscles, and we also want to then see if we're going to get the expression that we'd expect. So basically, the first thing that it's doing, can we actually monitor that? So two things that we look at here. One is going to be, can we actually quantify, can we measure the amount of the drug that's getting into muscle? And so that's what this is here on the top, vector genome copies per nucleus. We know that we need at least one full copy to be able to make microdystrophin because that's going to be a copy of the full gene. And more can only be a good thing in our case here, where we're really looking to see, can we both have robust levels of expression, and then also can we have durable expression over time? And that's what we're again encouraged to see these numbers quite similar between both that earlier time point and then also that later time point of 1 year. When we take the next step from here, we look to actually see the expression of SGT-003 microdystrophin. So that's actually looking at the protein itself. On the left side are some representative images here at day 90, and then the top is baseline. And those red circles here are actually going to be staining for dystrophin. And in our case, the baseline is going to be that what we'd expect to see for a patient who has Duchenne, which is really negative for dystrophin. And then at day 90, post-treatment, what we can actually see is that we have quite robust microdystrophin expression here. And this is as a result of SGT-003. So really seeing that across a majority of the muscle fibers. And so that's what each one of these red lit up circles are. And when we, again, quantify this, we can see that across multiple measures, this is using Western blot, which you may have heard of, and then mass spectrometry. These are ways that we actually measure how much protein do we have. So we can see that we have quite high levels here at the day 90 time point, as well as the day 360. So by these two methods, 60% of normal at the day 90 for Western blot, and then 52% by mass spec, and then also high levels out at day 360 as well. What What's important about this, and I always like to highlight this, is going back to that image. It's one thing to make a lot of protein, but you also want to make sure that it's getting throughout the muscle so it can really protect as many muscle fibers as possible. And that's where we're happy to see that the numbers here are on the majority of muscle fibers. So we have 63% of positive fibers at the day 90, and here 69% at day 360. So again, getting high levels of microdystrophin expression, and that expression is really throughout the muscle rather than being focused on just small areas. The next thing that we can look at when we're actually trying to see what are the biomarker changes that might be actually seen in the near term and then what can be monitored longitudinally are going to be some of these circulating markers, which we characterize as ways to really look at muscle integrity. And so I think all of us know serum CK very well, so creatine kinase, which is a marker really of seeing that the muscle membrane is fragile and that upon injury, you're going to see that CK is leaked into the circulation. So that's really why it's high at baseline in patients with Duchenne. And what we can see encouragingly is that post-treatment, both at day 90 and then here out to 1 year post-treatment, we see that we have quite substantial reductions in the CK that's circulating. So kind of an initial sign that the microdystrophin is being expressed, and also it could potentially be protecting the muscle from damage as we can see through these biomarkers. We also look at additional biomarkers because we really want to see if there is both a comprehensive story that we're seeing here that makes a sense from a consistency standpoint point of view. And so this is where we look at ALT, AST, LDH, and then another marker that's a bit more unique, which is called Titin. So we're looking at fragments of this large protein. And what we can see as we look across this here is that we're really seeing the same story across all of these biomarkers that are telling us after treatment, we're seeing less of what is normally in muscle and that would be released upon damage. We're seeing less of that in the circulation. So again, potentially seeing protection in these muscles. And so over time, we'll look to see functional results as well, as I mentioned, but the biomarker results have been quite encouraging for us and really motivated us to move from our initial phase 1/2 study to that next step of really wanting to characterize our functional changes in a controlled manner. And that's really where Impact Duchenne comes in. And it is a phase 3 study with that real key focus of evaluating functional changes. Because this study, especially, is newly ongoing and active, I'll go through a few more of the details here so that we can really know what the eligibility is for the study and what our plans are as it moves forward. Uh, so to date, we have 2 active clinical trial sites for Impact Duchenne. I'm very happy to say that one of those 2 is actually in Canada. And so this is working with BC Children's. And we're very happy again with the excellent teams, and we're looking to expand in Canada as well. The additional sites that we have active is over in Australia and Sydney. So another amazing team. And as we look at this, as I have here in the blue box, not only are we going to be adding sites in these locations, but we're also going to be expanding this study into the EU, into the UK, and the US. These are all going to be pending regulatory approval. So these are our plans to add these sites, but really hoping to be able to, again, impact as broad of a community as possible. Because the focus of this study is to be able to evaluate efficacy, the design is a bit different than the phase 1/2 study. Here, we're using a double-blind, placebo-controlled design, and patients are going to be randomized 1-to-1, either to SGT-003 or to a placebo. And this is really so that within the period of the study, we can more effectively characterize what the changes in function are based on the drug itself. So that's the real key piece here for a functional efficacy evaluation. We're still using the same approach as I mentioned for the phase 1/2 study. And so this is a single dose of SGT-003. We are using a primary endpoint at 18 months when we're looking at our functional efficacy. And I want to give a little bit of detail to this as well, because I think it's important. It's always going to be a goal of ours to be able to both look at efficacy changes as quickly as possible and also want to minimize a placebo-controlled study, especially as much as possible. We know, however, that in Duchenne, changes can take time. We're not always going to see immediate changes, especially when we're trying to control for some of these functional tests, which are not always going to be as sensitive as we want them to be. So our thought really is that the most important thing as we put this study forward is that we're not going to miss on the ability to see a treatment effect. And really, that was a key piece for making this design choice to be a bit longer than some of the studies have been in the past. But some of the studies also have not actually shown functional efficacy. And one of the concerns is that it may have been because they were too short. And so we do want to look at this and be able to evaluate what we think could be a better time point and also not going too much longer when we're looking at this functional efficacy evaluation. Also importantly, at this time point is when we're going to actually, those who have received placebo in the first part, once they get to 18 months, they will have the ability to receive SGT-003. So we consider this a bit of a crossover. So the only thing we'll look at is really safety evaluation. So want to make sure that the patient remains still eligible from a safety perspective. But if that is the case, those who receive placebo will receive SGT-003 at that point. So that's an important thing to highlight as well. For this study, we're going to be enrolling patients with Duchenne who are age 7 to less than 12 years. This is looking at an ambulatory functional endpoint. And so the functional status is required to be ambulatory. And there are some other functional criteria that are also going to be reviewed at the point of determining eligibility. I'll go through each of these items in a bit more detail since we have time here. The first, just to make sure that it's clear, is that because, as I mentioned, that potential for a myositis, the potential for an immune reaction against microdystrophin, we do have an exclusion criteria for specific genetic mutations in the DMD gene. And these are deletions only. So if you're looking at each of these exons here, so exons 1 to 11, 42 to 45, or 57 to 69, these are the ones that overlap with our microdystrophin. So if someone were to have a deletion in these pieces, essentially their body would have never had the ability to make a version of a protein that would give them a little bit more of a protection from an immune response against this. And so that's really why the exclusion criteria are necessary. But I want to emphasize again that this is only deletions. So if someone were to have a duplication or a point mutation, those would not be excluded. So always want to make sure to speak to a clinician, to an investigator about these specific details. But I do want to make that clear up front that just deletions are excluded. And then because this is an AAV gene therapy, we do test for AAV antibodies. And so this is something that will be done in the context of the study and must be negative so that we can essentially hope to have that treatment effect because antibodies would otherwise prevent that from occurring, which is also why we can't have a history of gene therapy because you would also have that antibody response and you'd be a little bit more of a variability than what we're looking to be able to characterize our treatment effect here. And again, towards that, wanting to reduce variability, treatments such as exon skipping therapies, vamorolone, givinostat, there must be at least 6 months of a washout period, so meaning that you haven't used that drug when you are going into the study. So if you were to be on this, one thing that I'll also call out is in our study, we allow for looking at antibody levels at the very beginning of the study. So this is an important part, especially if anyone is considering wanting to stop any of these treatments that they may currently be on. Having that information, and again, talking to a clinician, talking to an investigator about any decision like that. We also require stable daily oral steroids for 6 months. And then again, similar to what could be either approved or investigational therapies, would not be allowed within 6 months of dosing. In this study, as I mentioned, we're looking at an ambulatory functional endpoint, and that endpoint is time to rise from supine velocity. And so this is really time that it takes for someone to, from laying down on the floor on their back, to come up to standing. And this is something that we hope could be a bit more of a sensitive test compared to other outcome measures that have been used in the ability to actually see, again, that treatment effect at this time point, which is day 540 or 18 months. We'll also be looking at many additional markers here, and these are just some of our key secondary endpoints because we want to characterize the effects that we're seeing as well as possible. And so from an ambulatory functional standpoint, we'll look at things like using wearables to do stride velocity calculations, looking at the 4-stair climb test, the North Star ambulatory assessment, and other time function tests like the 10-meter walk run. So really wanting to get a very good idea of what our drug is showing in this placebo-controlled setting. In addition to these, we'll look at many other outcomes like pulmonary function tests and cardiac endpoints because we want to really get a good idea of both the efficacy and the safety and see over time how each of these are being impacted. And so, with that, I want to say thank you to everyone who's been listening today, everyone who'll listen in the future. Very much thanks to our clinical trial sites, but most importantly, everyone who's been involved in our studies, the patients themselves, the families, everyone who might be in any way associated with all of that, because these are such big commitments that are being made. So we really, really appreciate all of that time, energy, and effort. If there is any information that is needed, each of these are our clinical trial postings. So these are QR codes that go to our ct.gov postings for both Inspire Duchenne as well as Impact Duchenne. And you can also search those clinical trial IDs that we had earlier in the deck on ct.gov to be able to get to those for more information. We'll continue to keep up the community up to date on all clinical trial site activations that we're doing, especially. And this is the best way to find that up-to-date information is going to these locations. So again, thank you so much.
Thank you so much, both of you. That was really informative and really well put together. I do want to just let everybody know that I've realized the chat seems to be disabled. So if anyone has any questions, if you could go to the More button at the bottom of your screen and just select that, and then there should be an option to click on an icon that says Q&A, and then there you can type in any questions you have. And while you're doing that, there were a couple of questions that had been submitted ahead of time. And one of them related to challenges in the other trials. And I guess in thinking about future trials, what do you think your biggest challenge ahead is?
Well, that's a big question. So appreciate the ability to go through it a bit. No, I think the most important thing for us, especially as we look to characterize this drug for the first time, I think is not necessarily a challenge, but making sure that we are able to enroll enough patients, that we are able to really do our part as a sponsor to be able to provide what's necessary for all the families that are involved. And so, especially as we're starting this study out, I think we want to make sure that we're giving as much information to the community as could be helpful and could be necessary. So information is always a big piece of this, and we want to be as helpful as we can there. I think the most important thing is, especially within the context of a placebo-controlled study, as we have our Impact Duchenne study, it's a big decision to be able to commit to that. So I think it's very important, again, to talk to your clinician, to talk to your to the investigators for a study, to make those sorts of decisions. And we're hopeful that we'll be able to help along the way with anything we can provide for that as well.
Perfect. And then another question that came in, I think has more to do with just on future enrollments perhaps, but says any visibility on future enrollments for Inspire Duchenne, knowing that 46 kids have already been infused.
Yes, we've been really privileged again to have quite substantial enrollment over the last not even 2 years where this study has been going. As of this point, we are looking to conclude the planned enrollment that we have for to date in that study. If there are any changes to come, we'll be sure to again update the community on that. But as of right now, just in these under 12-year-olds, we are looking to kind of make that transition from that open label study over to the phase 3 study for the time being.
Perfect. And how will you be protecting placebo patients against antibody exposure within the impact community? Is there a higher risk that they won't be eligible at the 18-month mark?
That's a really good question, and it's an important piece to think about in a placebo-controlled study. I think in general, what we always want to say is this is, again, a conversation to have on, what kind of precautions might be appropriate. This is not necessarily going to be recommended to, go into a bubble and not be able to interact. So I think it'll always be on a family-by-family basis to be able to make what's the right decision. We're encouraged to have seen some prior studies where we have had little to no seroconversion between a placebo-controlled start and a crossover time point. That doesn't mean that we won't see any, so we always do have to call it out as an appropriate risk to consider. But again, hoping just to have some standard precautions to be able to maintain that over the course. Annie, anything additional you might want to add?
Exactly that, Pat. I think it really comes down to what is doable for a family. We're not going to try to cause any additional burden for folks. And clearly, this decision was made in an informed way and also incorporated feedback and insights from patient representatives and advocates from around the globe. So, yeah, no, I think that really answers that.
We have another question that came in. Do you believe treating very young patients with limited fibrosis could significantly improve the long-term functional impact of SGT-003 compared to treatment later in disease progression?
I love that question. I think it goes back to thinking about, what's the mechanism of this treatment? And so ultimately what we are doing is we're looking to restore dystrophin in muscle that is present. And so I think there's a lot of great potential that the more muscle you have, the more potential benefit you could also have over time. And so we'd be protecting more at that point. I think what's important for us is to make sure to protect whatever muscle that you do have at that point in time, though. So, different patients are always going to be at different stages. And, if we're going to have the broadest impact, we want to make sure that we can get to as many as possible. So I think the potential is there. We've been really fortunate to have our open label study go down to dosing as young as 1 year old. So I think it'll take a little bit more time to see what those impacts are because we don't have all of the same tests at the younger ages that we can do as we do in some of the older boys. But there's good potential, and so we definitely want to protect muscle whenever we possible.
Excellent. All right. And then there's a question about recruitment of international patients. And I wondered if you could maybe just speak to some of the sites again that you're at.
Yeah, so right now we have 2 sites. We are planning to expand that quite to a quite high amount. But what I'll say is it'll always be an individual site's decision on whether they will be allowing international participants to be going to their site. So I would communicate, I would say for anyone to reach out to an individual site, see if that were to be a policy. It's not always going to be the same on every day as well. Some, conditions may change. There may be reasons for why international enrollment may or may not be feasible for a site at a certain point in time. As a sponsor, it's something that we allow and we do support. So if it is feasible at an individual site, just like any other trial participants who would be coming, cost would be covered and things from that side would be supported by us. So always, again, something to communicate with the site directly, and those site lists will keep being updated on our posting, if not also in other webinars or other forums like this as we have more.
Oh, yeah. And we're happy to share all of that as well with our community.
Great.
All right. We have another question. How durable, oops, where did it go? Hang on, it just jumped. How durable do you currently expect SGT-003 expression and function benefit to be over time?
Mm. It's something that we're going to always take time to learn on our own. We can point to other studies that have existed where we see kind of promising results that over years there can be treatment effects that are also seen. In our specific study so far, we've looked at about 1 year post dosing, and now we're starting to get a little bit later than that. Those are the 2 biopsy time points that we've included. And so that's where at least we can see that we have that expression that's maintained. And then also the biomarkers that I highlighted were out to 1 year as well. So we've seen that consistent response and improvement at that point. But time, I think, will tell for us. So we generate our own data to be able to support the exact amount of time. Our hope, again, as we know that this is a single time therapy, that will be able to characterize those longer term effects and hope to see that.
Perfect. And do you believe early treatment could eventually reduce long-term dependence on steroids by preserving muscle before significant damage occurs?
Oh, another great question. So, when we look at our therapy, we are doing it in the context of standard of care, which is steroids. So, that's why patients who are enrolling will be on that stable steroid regimen. Ultimately, I think that's a similar question to the durability question and understanding really, both what's the magnitude of effect that we'd see from our treatment, and then also how long will that effect go out to. So, I think it'll take some things like more real world settings to be able to evaluate those kinds kinds of questions over time, and then we'll get some of these better answers. But at least to date, we want to make sure that we're maintaining a standard of care, and then kind of building on that, and then making any other sorts of questions or investigate those sorts of things just as we learn more. But
Perfect.
I love that potential. So it's always great to call out.
Yeah. Okay. And then we have another question that came in that's just asking for your opinion on what you feel is the best available technique or method to control the disease so far.
Well, I would say the best method is to talk with your individual clinician about what's right for you. So, won't give a kind of a stance here. I think we understand again what standard of care is. So that I think has at least been always well characterized. But everyone is going to be different based on your specific age, based on your specific situation. So there's no one size fits all. I think we know that quite well for Duchenne as well as for many other diseases. But I think the most important thing is to really investigate that with your own clinician. And we are learning a lot more, and there's a lot more both clinical trials as well as drugs that are becoming approved. So it's becoming a bigger conversation and a bigger question to go through, which I think is a good thing to have more options out there. But it'll always be individual to you. That's most important.
Perfect. All right. And that one other question. What is the impact expected? When is the impact expected to take place in the US? So if you have dates at all.
Yeah, so I can't share a firm date right now. We are in the process, as we are with the additional regions that we're looking at expanding into, of really working with the regulators and getting things like site activations in place. So we hope that it's not going to be in the not too distant future for when we can start to make some of those updates. But we're currently in the process of actually trying to bring US sites online.
And as
Perfect.
and when those sites do come online and are activated, we will share that with the community in multiple ways. It'll be via community letters that we'll share with Defeat Duchenne Canada. There will be additional webinars. And as Pat kindly mentioned earlier, that information will also be available on clinicaltrials.gov.
Perfect. And we can share that on our website as well. Two last questions. One is delivery of treatment an outpatient day?
Yeah, I appreciate actually getting asked the question.
Mhm.
Yes, we are administering this as an outpatient therapy. And so that's a good thing to highlight. I think, especially based on our progression throughout the study, and now having more patients who have been dosed, we have a bit better of an understanding of the safety profile, that it has been quite consistent, which is always encouraging because I think the biggest thing that you can be concerned about is when you have variability, you don't know what to expect. So it's very important for us to see that things are consistent. So because of that, we have maintained the ability to have outpatient dosing.
Perfect. And the last question is, obviously this medication is being tested on individuals under 12. The question is, are there any clinical trials available for 18-year-old boys in Canada that you're aware of?
That's a great question. So for our program specifically, as mentioned, we do not have an adult cohort that is currently open. I think the best thing to do would be to go to clinicaltrials.gov to see if there were any other programs that may have cohorts that are enrolling that are in that specific age range.
Absolutely. And also, if you haven't, for those on the webinar, if you haven't seen our clinical trial page, if you go to the research tab on our website, you'll be able to find a clinical trials tool there and get that information as well. Actually, do we have time for one last question? I think so. Okay. Based on the data observed so far, what gives you the most confidence that SGT-003 could provide durable long-term clinical benefit? I think that's a very
Yeah.
similar question to the one we had before.
It is, but I think it's important to highlight. Yeah, when we look at things, especially that are going to be closer to the time of treatment, I think those are initial effects that we hope to see changes in, but it is important to look at later time points as well. So, especially now that we have the 1 year results, because we have more of this, again, consistent profile, I think that's what's been most encouraging. We
Mhm.
don't look at just one marker, we do look at many so that we can see, are things moving in the same direction? Are they telling that same story? So to me, and I think to us, that's what has been most encouraging is looking at not just day 90, but now 6 months out, 1 year out, we're seeing that same sort of a picture being displayed. So encouraging start. We have a lot of work to do as we continue to evaluate this. So I'll always call out that disclaimer, but that's exactly why we're running this specific study for Impact Duchenne.
Perfect. All right. Are there any more questions out there?
I have one question, if you don't mind.
Yeah, go
Hi,
ahead.
Jenny, and hi, Pat.
Hey,
So,
Nicola.
so great to see you both, and thank you so much. This was an incredible, informative presentation. I really appreciate it. My question is mostly just for Canadian families who are interested in participating in this trial. So, it's my understanding that there's one trial open in Canada at the moment. I mean, Canada's a very large country. Are families who may be interested in other provinces, is it possible for them to travel to a trial site if they're interested, or how would they get more information on how to do that?
Yes, absolutely. So we are looking to expand geographically as well with the sites that we'll have included in Canada. So hopefully, again, more to come on the updates there. We would also support travel for anyone who is traveling within Canada to a site. Again, one thing that we want to make sure is to work with you all very closely so that any information that we can provide, any questions that might come up, we can be sure to answer. But that is our hope, is to be able to cover much more of the overall very large area of Canada, so that it won't be just the one location on the West Coast.
That's good to hear. Excellent.
Thanks, Pat.
Thank you.
All right. Last call for questions. All right. So every time we do a webinar, it's really important to us that we seek your evaluation so that we know how to grow and improve for other webinars. And of course, we will share the evaluation with Solid Biosciences as well. So if you'd like to take a screenshot or just use the QR code to pull up the evaluation, we encourage you to complete it as soon as possible. And then we'll also send you one to everyone who has registered as an attendee today. So you will get that in an email later today. Thank you. Next slide. And just wanted to put the plug out there that we're on social media. We're on Facebook and Twitter and all different places. So if you haven't seen us on all our channels, please join us, and we'd love to see you there. Next slide. And lastly, we also have an e-newsletter that you can sign up for. So we encourage you to do that if you haven't already done so. And that is really it for today. So I want to thank everyone for coming today. And I want to give a huge thanks to Solid Biosciences for not only their support, but their amazing information that I know is really important to our community. So I cannot thank you enough. And it was a wonderful presentation today. Thank you.
Thank you so much. Thanks, guys. Appreciate it.
Thank you.
Thanks,
So appreciate
everybody.
it.
Thank you.
Thank you,
Bye.
guys.
Thank
Have a
you.
have a good afternoon, everyone.